Today's video is all about some additional accessory structures that some bacteria have that are also external to the plasma membrane. So we're going to talk about a glycocalyx, which is also called a capsule, and then flagella and Fimbriae and pili, again, all of which are accessory structures. So not all bacteria have them, but some do, and they normally offer the cell some kind of advantage. So first up is the glycocalyx, and the glycocalyx is a sugary coating, and we're not entirely sure. Sometimes they include proteins, sometimes they don't, but mainly just remember that it is a sugary coating that will protect the cells, and sometimes the glycocalyx also helps it adhere to surfaces present in the environment. So different bacteria have differences in their glycocalyx, if they even have one. And the capsule is a type of glycocalyx that is very thick, and it is bound very, very tightly to the cell. Okay, and there are staining techniques that we'll talk about in lab that we can view them. So a lot of pathogenic bacteria have a capsule or a glycocalyx, and that is because the glycocalyx is considered a virulence factor. It is a strategy that pathogens use to evade our immune system. Specifically, capsules help protect the bacteria from phagocytic cells. Remember, phagocytic cells, their purpose is to go and engulf foreign invaders and digest them to get rid of them. And so this sugary layer makes it really hard for phagocytes to grab hold of these organisms and eliminate them. Okay, and so when these bacteria are able to escape phagocytosis because of this sugary capsule, they are then able to replicate within the body and infect various body tissues. Okay, and there have actually been studies that show that the same type of bacteria that does not have a capsule, like if we've genetically manipulated them in the lab to study them, and we've made it so that a bacteria does not have a capsule when it normally would, they actually lose their ability to be pathogenic, which just really goes to show how important capsules are as a virulence factor. Another important role of the glycocalyx is the ability to form biofilms. And so biofilms can form in all sorts of environments. For instance, white plaque that forms on the teeth. Sometimes that's in part from organisms that have a glycocalyx. And it also helps protect the sugary structure. The capsule helps the biofilm remain adhered to teeth, so it doesn't just slide off as we talk. And as I've mentioned in class, for those of you that have me in lecture, they can be really challenging in a hospital setting because the sugary capsule allows them to adhere so firmly to structures that they can actually result in persistent colonization on non-living materials like catheters and pacemakers. Okay, so they can essentially clog those and wreak havoc on the human that they're inside. So let's talk a little bit about biofilms, which is the picture in the top left. And so you can see biofilms in other areas like your shower, even your toilet, and like I said a second ago, on the teeth, or in medical equipment. So it turns out that a lot of bacteria don't like to live in what's called a planktonic form, which is essentially as a single cell, a lone ranger by itself. It turns out most bacteria like to live in these cooperative communities full of various other organisms, including algae, fungi, things like that. And the reason biofilms are advantageous to these organisms is because these biofilms are essentially little habitats that provide plenty of access to, water and food and other factors that these organisms need. But they also cover themselves with this like extracellular matrix. Okay, and that's what you see is like, it looks like the green coating in the bottom of that picture there. And that actually protects them as well from things like antibiotics or antimicrobials, because it's this thick extracellular material that the antibiotic must penetrate to even get to the bacteria or whatever species or type of microbe that we're targeting. And so biofilms are very advantageous for the bacteria, but they make our human lives much more difficult because they're harder to kill. And again, they like to grow on things like, IUDs and shunts and urinary catheters and things like that. So it can be pretty problematic. Okay, next up, let's talk about one of my favorite accessory structures, the flagella. I love them. All my favorite things end up on quizzes and exams, so you definitely want to know all about flagella. Flagella are used for movement or motility, okay? And in bacteria, they actually function like a propeller, okay? Like think of like maybe a boat propeller or even like a helicopter propeller. And these flagella are rooted, okay, to the plasma membrane. And that's what these top pictures are showing you. I'm not going to ask you to label any diagram like this. I'm just trying to show you that flagella are always firmly anchored to the cell, whether it's gram-positive or gram-negative. Now, bacteria vary in their flagellar arrangements. Some don't have flagella at all, okay? So trichus, a student helped me out and told me that trichus is a Greek word for hair-like, because these flagella do look like hairs, right? So if you see the word root trichus, then you know we're talking about flagella. So bacteria that don't have any flagella are said to be atrichous because the prefix a means without. So that would be without hair-like structures. So at the bottom, you can see the different types of flagellar arrangements. Okay, monotrichous means there's one single hair. When there's one at both ends, we use the prefix amphi. Okay, so amphitrichous means there's a flagella on both ends of the bacteria. Lophotrichous means there's like a tuft or a clump of flagella at only one end of the bacteria. And then peritrichous means that the organism is completely covered in flagella. And I remember this one because to me, these bacteria look hairy. Well, what rhymes with hairy? Peri. So peritrichous is hairy looking and covered in flagella. So as I mentioned, flagella, when they're attached to bacteria, move like a propeller, okay? So in this figure, they are moving in a clockwise position. And we say that bacteria run or tumble, okay? So when they're moving nice in a straight line, we say that they are running towards whatever it is that they're moving towards. But when they need to change direction, they're suddenly trying to get all of these flagella to move in the opposite direction. So it's not a very graceful process, okay? Imagine when you have your arm under the water, like in a pool or a river or lake or whatever, and you're trying to move your hand real fast, right? It can be hard enough to move your arm quickly underwater, but now try turning it real fast the opposite direction, right? There's a bit of resistance. It's hard to do. So that same type of resistance is there for all those individual flagella. So they're not very graceful when they're turning. So ungraceful, actually, that we call it a tumble. They just kind of sputter around trying to change direction. If you have time, I do suggest you look up, run and tumble like on YouTube, look at flagella motion and seeing how bacteria move. I'll try and find a video of that to post for y'all, but it's pretty snazzy. Okay, for the sake of time, let's keep going, but please definitely be familiar with those flagellar arrangements and how they work and the terminology associated with bacterial movement, run and tumble. All right, our last slide talks about two different accessory structures. So again, not all bacteria have them. The first one, or the first structure, they're called Fimbriae. And these are tiny, short, hair-like projections that extend from all over the cell. So if you look carefully at this picture at the top cell, if you look real close, you can see there's a lot of little tiny hair-like projections completely covering that top cell. Those are Fimbriae, okay? And they completely cover the cell, and they are used for attachment, okay? They help organisms attach to various structures. They also tend to be virulence factors as well. A lot of a lot of pathogenic bacteria tend to have Fimbriae because it facilitates attachment to their host, which means it's easier to help them get sick or make the host sick. Okay. Next up are pili, and pili are found in certain bacteria. We're going to talk a lot more about pili in future videos, but these are long appendages. Normally there's only one or maybe a few, and they do aid in attachment, but it's cell-to-cell attachment, okay? Because it turns out a pili has a role in helping cells exchange DNA. So the cell that has the pili will essentially extend this pili outward. It's kind of like fishing, right? When you cast your line outwards. So the cell that has the pili extends that pili outward. It literally elongates it until it attaches to another cell, and then it reels that cell in. It brings that cell closer to it so that it can then exchange its DNA. Okay, it's a more complex process than we need to talk about right now, but just know that a pili are involved in DNA exchange or the transfer of DNA. Okay, we're talking a lot more about that later. All right, that is the end of the external structures on prokaryotes. Let me know if you have any questions.